Hello children, in today's video number 3, I am going to deal with meiosis. So before going into the details of meiosis, let us take a recap of the different stages of meiosis. We know that meiosis is of very long duration. It comprises two subphases, mainly meiotic division 1 and meiotic division 2. And meiotic division 1 in turn comprises the 4 stages: prophase 1, metaphase 1, anaphase 1, telophase 1. And prophase in turn comprises 5 substages: leptotene, zygotene, pachytene, diplotene, and the last stage diakinesis. Now going into meiotic division 2, it comprises 4 stages: prophase 2, metaphase 2, anaphase 2 and telophase 2. So it is very lengthy, having many stages and substages. The 2 phases are meiotic division 1 and meiotic division 2. Meiotic division 1 in turn is divided into 4 stages: prophase 1, metaphase 1, anaphase 1, telophase 1. And prophase 1 is in turn divided into substages, namely leptotene, zygotene, pachytene, diplotene and diakinesis. And meiotic division 2 is in turn divided into 4 stages: prophase 2, metaphase 2, anaphase 2 and telophase 2. So I will indicate in white. This is the phase, these are the stages, and only for prophase there are the substages. So let us get started with meiosis. So let us start. Definitely when we start cell division, we have to start with the first phase from which it starts off, that is the interphase. And we all know interphase is a stage during which the cell is getting prepared for cell division. And in this stage we see the nuclear membrane is intact, the nucleolus is intact, and the centrioles outside the nuclear membrane is also intact, and the chromosomes are in the form of fine entangled thread called chromatin. Now meiosis and mitosis, this is the state of interphase. But in meiosis I am going to depict the chromatin with 2 colors, red and blue. You also draw it with 2 colors, red pen and blue pen. So I am erasing the chromatin that is depicting the mitosis, which is with the normal pen or pencil. I am using 2 colors, red and blue, red and blue color. Now as I started off interphase in mitosis, can you identify the number of chromosomes in the interphase stage here? How many number of chromosomes are there? Can you guess? No, but always we go with 4. So from this you should understand there are 4 chromosomes. Of these 4 chromosomes, 2 are red in color and 2 are blue in color. Okay, we are depicting 2 chromosomes in red and 2 in blue, but because they are elongated and entangled, you cannot identify the start or ends of these chromosomes. It appears as an entangled mass called the chromatin threads. So to start off with meiosis, we have to first describe an interphase stage. Interphase stage, as I told you, like any other interphase stage, is the stage in which the cell is preparing for cell division. Here the nuclear membrane is intact, the nucleolus is intact, the centrioles are intact, but the chromosomes, how are they? They are elongated in an entangled mass called chromatin. And remember, in meiosis, what I have done is I have taken two colors, red and blue, and showed the entangled chromatin. whereas in mitosis I am going with one color. Here it is required that you have two colors. So let's get started. The interface, as I described, the nuclear membrane is intact, the nucleolus is intact, and centrioles outside the nuclear membrane is intact, and the chromosomes are in the form of chromatin threads. Now we are moving on to prophase. So I am sliding this. We are moving into prophase, and prophase, as I told you, is divided into 5 substages, and I hope you can see all 5 substages displayed on the template. First substage is leptotene, second zygotene, third pachytene, fourth diplotene, and the last substage is diakinesis. Now here it is very easy. When you are drawing, you just draw the square and the circle right across till the end. 5 squares with 5 circles inside indicating the cell wall or the plasma membrane, the circle indicating the nuclear membrane. And here we are noticing only one event occurring, the event occurring with respect to the chromosomes. So first, from the interface stage, we are proceeding to the leptotene stage. What has happened? One event. The event is condensation of the chromosomes to form rod-like structures. Once again, condensation of the chromatin to form rod-like chromosomes. See, I have drawn here. Look at this picture. The 4 chromosomes, rod-like chromosomes, and be very careful when you draw these rod-like, 2 reds and 2 blues, and they have to be scattered. Don't copy the same orientation, alignment as I have given. You can draw 4 rod-like structures, 2 in red and 2 in blue, scattered. And what is the event is condensation of the chromatin to form chromosome rods. Clear? Next event, from leptotene we are moving to zygotene. What happens here? From the diagram itself you can understand pairing of similar chromosomes. Here similar stands for homologous chromosomes. So the blues together pair and the reds together pair, and they pair to form a bivalent. So the event occurring in zygotene stage is pairing of homologous chromosome or pairing of similar chromosomes to form a bivalent. What is a bivalent? 2 similar chromosomes constitute a bivalent. So we have 2 bivalents, one in blue and the other in red. Here also orientation does not matter provided you give it in pairs. You pair 2 reds and 2 blues, put it anywhere within the nuclear membrane. Clear? So this process is called synapsis. The pairing of homologous chromosomes to form the bivalents is called synapsis. Next, from zygotene we are going to the pachytene stage, very, very important stage, pachytene stage. Here we can see this bivalence further condenses. To form the bivalence further condenses, it becomes thick, and we can see the chromosome visible as X-shaped structures. So if I display it separately, see, bivalent, this is one blue and another blue chromosome. This is representing the chromosomes at the zygotene stage where they are rod-like, and not only that, they are paired to form a bivalent. Now they further condense. When they further condense, you can see the X-shaped structure. Now this we referred to as bivalence. And now when it condenses further, it becomes thick and visible as X-shaped structures. This is referred to as tetrads. So as a result of condensation, the rod-shaped chromosomes of the bivalent condenses, therefore it becomes thick and forms the tetrads. Clear? So when you count, you can see it is a tetrad. 1, 2, 3, 4. 4 arms. So it's called a tetrad. Here it's just 2 arms. 1, 2. So it's called a bivalent. So one event occurring in pachytene stages, further condensation of the bivalents to form tetrads, and the second event is they cross over. Now again here I bring attention to these terms. This is one chromatid, this is the second chromatid, and these chromatids are connected by a point called the centromere. You are familiar with this. And the name of these chromatids are sister chromatids. These are sisters because they belong to the same chromosome. So, they are called sister chromatids. Okay. Now, I am drawing the other homologous chromosome in lighter color. One sister chromatid, the second sister chromatid connected by the centromere. Okay. So, these two are sisters. Now, what is the relationship between this light blue and this? These are non-sisters. So, this is a non-sister and this is also a non-sister. Okay. And these are adjacent non-sisters. You have to address this term adjacent non-sisters because in Pachytene stage the next event is dealing with crossing over. So, I am going to show crossing over here. The chromosomes, see, it crosses over. So, the next major event that is occurring in Pachytene stage is the crossing over between the adjacent non-sister chromatids. I told you what are the non-sisters, and remember it is between the adjacent non-sister chromatids. So, once again, if I bring this back, so the event that is occurring, the first event is condensation of the chromosomes. From rod shape they become visible as egg shape. So, the bivalents become tetrads. So, that is the first event. If I am indicating in pink, this is the first event that is occurring in Pachytene stage. The bivalence condenses to form the tetrads. I hope you have understood. These rods further condenses, you get the tetrad. This is the first event occurring in Pachytene stage. The second event occurring in Pachytene stage is, I am going to show it in action. The two chromosomes, that is of the bivalent, the two homologous chromosomes, they cross over. See, I crossed it over. Now, they cross over, see, and see this region, please note this region, to form an egg-shaped structure. This egg-shaped structure is known as chiasmata. So, what is the second event occurring in Pachytene stage? The second event is called crossing over. What is crossing over? It is the non-sister chromatids, that is the adjacent non-sister chromatid crossover to form an egg-shaped structure called chiasmata. Okay. If this is one non-sister, this is the other non-sister, they come close and cross over. So, we have one chiasmata here and one chiasmata below. So, these are the two events occurring in Pachytene stage. So, what is the event occurring in Pachytene stage? Remember the two C's, C and C. The first C is condensation of the bivalents to form tetrads. Got it? The bivalence of the homologous chromosome becomes tetrads. Then second event is crossing over takes place between the adjacent non-sister chromatids. So, that is occurring in Pachytene stage, and this, see how I have depicted Pachytene stage, have a closer view. And now, did you notice that in addition to showing condensation and crossing over, what I have done, the blues itself, I did not take two shades of blue, it will be more confusing for you. Instead, I made one blue chromosome thin and the other blue chromosome thick. Similarly, with the red chromosome, one thick and the other thin, because the next event is based on this. What is happening in diplotin? In diplotin stage, what happens is this chiasmata breaks. With green I am showing this chiasmata breaks here. So, it breaks here and here also it breaks. Now, what happens when it breaks? It breaks and separates such that the chromosomes get their exchanged parts. So, you can see the thin, see this is a thin blue chromosome, now I have highlighted in green. It has thin parts, but one portion is thick because it is of this exchange, it has exchanged with the thick blue. Now, same way the thick blue chromosome, 3/4 is thick, but you see that exchanged part is thin because it has exchanged with a thin. So, this is very, very important in Pachytene stage and diplotin stage. Not only you are indicating colors, you are indicating also with respect to the thickness. So, once again I am reiterating, when you draw Pachytene and diplotin stage, you see that you draw in two colors, not only in two colors, within the two colors, one chromosome you show in thin and the other you make it thick, so that you can show crossing over and breaking of the chiasmata. After breaking of the chiasmata, the separated chromosomes have exchanged paths. Now you can understand from this diagram that the paths have been exchanged. The thick of this chromosome is exchanged here, and the thin part of this chromosome has exchanged to the thick chromosome. Clear? So that indicates diagrammatically the event. So meiosis should be explained to the examiner both in your diagram as well as in your write-up. It should be expressed. So remember, in Pachytene and diplotin stage, not only the two colors, you are also indicating with the thickness of the diagram, that is thickness of the nib. Clear? So once again, what are the events occurring in Pachytene stage? Further condensation of the homologous chromosomes of the bivalents, and they become tetrads. So the two blues further condenses to form the two X-shaped structures, which is forming one tetrad, and the two red bivalents further condenses to form the red tetrads. And not only that, this crossing over between the non-sister adjacent chromatids, between adjacent non-sister chromatids. See, these are the adjacent non-sisters. I have depicted when I showed that display. Okay, between the non-sister adjacent chromosome, this crossing over, and that crossed over X-shaped part is called chiasmata. That's very important to mention here, because when you're explaining about diplotin, what happens in diplotin stage is the breaking of the chiasmata. Once the chiasmata break, the chromosome separates from one another with exchanged parts. How do they separate? With the parts exchanged. Now moving on to the last, which is very, very easy. What happened is, again you need not continue with thick and thin, but you continue with red and blue colors. Okay, what happens in the last stages? It's depicted in the diagram. The nuclear membrane disintegrates, the nucleolus also disintegrates, and the centrioles splits and move to the opposite poles. See, it's on its way to the poles. Clear? So in diakinesis, what happened? Nothing related to the chromosome, but the other features. That is, the nucleolus disappears, the nuclear membrane is disintegrating and it will slowly disappear, and the centrioles duplicate or replicate and move to the opposite poles. So both diagrammatically and in writing, you have to express each stage. Very, very important are these 5 stages, because the diagram as well as the write-up should express what is happening in each stage. So once again, a recap of the events occurring in the prophase 1 substages. Once again, now I am not going to explain with the diagram, I am going to explain only the event. Leptotene, the event occurring is condensation of the chromatin to form rod-shaped chromosomes. Zygotene, pairing of the homologous chromosomes to form bivalents, and this process is called synapsis. Pachytene, 2 events occurring, remember C and C. Further condensation of the bivalents to form tetrads, and second event, crossing over between adjacent non-sister chromatids. Diplotin, breaking of the chiasmata, and the chromosome separates from one another. The chromosomes of the tetrad separate from one another with the exchanged parts. And lastly, in diakinesis, nothing happening with the chromosomes. What happens there is the nuclear membrane disappears, the nucleolus disappears, and the centrioles replicate and moves to the opposite poles. Now I emphasize only what should be written. Now the diagram part. Now the next round I am talking with respect how you have to draw the diagram. We started the diagram with interphase. See, we started the diagram with the interphase, which is represented like this. That is, the nuclear membrane is intact, the nucleolus is intact, the centrioles are intact immediately outside the nuclear membrane, and the entangled thread, chromatin thread, depicted in both red and blue color. This red and blue you have to continue till the end of meiosis. Now from there I proceeded to leptotene stage. See, in leptotene, what did I show? The rod-shaped chromosome, 2 reds and 2 blues, rod-like structure scattered. Then in zygotene, what did I show? I paired the 2 blues and I paired the 2 reds in my diagram. I am only explaining from the diagram point of view. Then in the Pachytene stage, what did I do? I showed 2 blue X's and 2 red X's, and not only that, among the 2 blue X's, one X I made thick, and among the 2 red X, one X I made thick. And not only that, along with those depictions, I made them close to each other, crossing one another, showing the chiasmata formation. And diplotein stage, what I showed, the chiasmata has broken and separated, the two homologous chromosomes of the tetrad has separated with exchanged parts. So what you have to draw is, you are drawing the thin blue with a small portion of thick in both the arms, and in red also you are showing the exchange parts. Okay, so you have to, this diagram should be in continuity with the Pachytene stage. Okay, there should not be any anomaly, it should be like a continuation. Okay, so from here, when you proceed here, you can understand chiasmata has broken and the chromosome has exchanged the parts. Then again, you need not go with thick and thin. The last diagram is the most simplest. Anyway, you have to depict the two tetrads, the blue tetrad and the red tetrad, and the nuclear membrane disintegrated in the form of dotted lines, the nucleolus slightly as a light spot, and the centrioles moving to the opposite poles. So I have explained in point of view from the diagram as well as from what you have to write. Very, very important, because both diagrammatically and in the write-up you have to express what is happening in each stage. Next, moving on to metaphase. Metaphase, what happens is that the tetrads align at the equatorial plane. So if this is the cell, the equatorial plane, like the equator of the earth, is the center. Okay, the horizontal plane at the center. So we can see the tetrads align at the equatorial plane. Now there is a common error for you all to make. There is always a mistake. When you think of tetrads, sometimes you might bring the tetrads like say two reds like this way and the two blues like this. It should not be aligned like this. What you have to do, it should be aligned like this. Be very careful of the alignment. This is the correct alignment, whereas what I am going to show is a wrong alignment. This is a wrong alignment, very wrong. Okay, so when you are saying that the tetrads align at the equatorial plane, this is the way you have to depict. The red tetrad and the blue tetrad align at the equatorial plane. See, it's aligned at the equatorial. Don't align like 4 at a stretch horizontally. No, this is wrong depiction. This is the right depiction. Clear? Now, meanwhile, they have aligned at the equatorial plane. So you saw the red tetrad, see, and the blue tetrad at the equatorial plane. Now, meanwhile, we have the centrioles at the poles. They give out spindle fibers, and these spindle fibers connects with the homologous chromosomes of each tetrad. See, they are connecting with the homologous chromosomes of each tetrad. Clear? Now, anaphase stage. So, remember the two events occurring in metaphase. One, alignment of the tetrads at the equatorial plane. Please give the proper alignment. And second, the spindle fibers arising from the centrioles from two poles attaches with the homologous chromosomes of the tetrad. Definitely, it is attaching to the centromere itself. Okay. Next is, in action you can understand what is happening. In anaphase stage, the centrioles give out spindle fibers. These spindle fibers contract, pulling the chromosomes to the opposite pole. So you can see in the tetrad of homologous red chromosomes, they separate. One red goes to the one pole, the other red goes to the opposite pole. Same way, blue goes to the opposite poles and the other blue goes to the down pole. Clear? So the contraction, so the event occurring in anaphase stage is the spindle fibers contract, pulling the chromosomes of the homologous pair or of the tetrad and separating them to the opposite poles. So the homologous chromosomes separate. One red and one blue goes to the upper pole and one red and one blue chromosomes goes to the lower pole. Okay, clear? Contraction of the spindle fibers resulting in the separation of the homologous chromosomes from one another. The red and red were together, now they separate and goes away. The blue and blue were together forming a tetrad, they separate and move away to the opposite poles. That is happening in anaphase stage. And next we are moving to the telophase. Telophase, like the previous what we have learnt in mitosis, reappearance of nuclear membrane and nucleopilus. So there is a nucleolus that reappears at the two poles. Say nucleolus reappears, which I have spotted now, and also the nuclear membrane also reappears surrounding the chromosomes. And already there are the centrioles. No, centrioles gave rise to spindle fibers, they are already present. Okay. If you want, you can continue with the same diagram. Let the centrioles be where it was earlier located, here itself. Clear? So what happens in telophase 1? Reappearance of the nuclear membrane which surrounds the chromosomes, and also reappearance of the nucleolus. So we have two nuclei in one cell. Now what happens is a binucleate structure is formed as a result of telophase 1. Now let us move to cytokinesis. As the name suggests, this cytoplasm constricts and divide the cell into two. So this very same diagram, only thing it is divided. The cytoplasm divides the cell into two. So we have a dyad of cells. Nothing has happened with respect to the nuclear membrane, nucleolus, centrioles or chromosome. The same diagram, only thing the constriction of the cytoplasm took place. It constricted sometimes and splits the cytoplasm into two cells. So we have a dyad of cells. And the last stage, what happens? What's the difference between the penultimate and the ultimate stage? You can see the chromosomes elongates and become entangled mass called chromatin fibers. So here they elongate. The red and the blue chromosome, they were condensed, now they elongate. When they become elongated and to get accommodated in that circle, they become an entangled mass called the chromatin fibers. Here also this takes place to form the chromatin fibers. So we get back the initial diagram, the interphase diagram, and this is a very short lived phase. This phase lies between meiosis 1 and meiosis 2, and this phase, which is similar to the interphase, is called interkinesis. It is a very, very brief phase between meiosis 1 and meiosis 2. There are two cells with two chromosomes. We start with 4 chromosomes, but now when you see, there are only 2 chromosomes in each cell, and these 2 chromosomes gets elongated and entangled, okay, to form 2 interphase cells. So we are having 2 cells, 2 daughter cells are produced. 2 daughter cells are produced. And remember, describing this daughter cells in the interphase, like any other interphase, the nuclear membrane is present, the nucleolus is present, the centrioles are present, and the chromosomes are in the form of chromatin threads. But remember, we started with 4 chromosomes, now it has reduced to 2. The same description to the second daughter cell. So let this daughter cell be A and this daughter cell be B. Now it is going to proceed to which phase? Meiosis 2. In meiosis 2, we have prophase 2, metaphase 2, anaphase 2, telophase 2, and cytokinesis. This meiosis 2 is exactly equivalent or similar to mitosis, but we are starting mitosis. We start with 4 chromosomes, but here we have to continue. So we have to continue with 2 chromosomes. Okay. So now each of the daughter cells, the A daughter cells is going to undergo all these phases, and the B daughter cell is going to undergo these phases. Okay. So if you recollect mitosis, we can complete this diagram. What happens in mitosis? I am drawing the cell: prophase, metaphase, anaphase, telophase, and cytokinesis. Exactly a recap of mitosis. But here also you have to use your red and blue pens. So we are having the nuclear membrane disintegrates. We have learnt in my nuclear membrane disintegrates, nucleolus disintegrates, centrioles move to the opposite poles. Okay. And what happens to our chromosomes? They condense to form rod-like structures. So we are having only 2 chromosomes, and one red, one blue. So, one red and one blue. Draw at random. Now, we are moving to metaphase. Metaphase, what happens? Further condensation takes place and the rod shaped chromosomes become X shaped, and not only that, it aligns at the equatorial plane. So, we have one X shaped chromosome there and one X shaped chromosome aligned at the equatorial plane. And meanwhile, the centrioles which have reached the poles, the centrioles I told you is on the way to the poles in prophase 2. Now, it has reached the poles and gives out spindle fibers which connect to the centrioles of the chromosomes. Next, anaphase stage, what happens? The centrioles, the chromosomes are being split by the centromere. The red chromosome is split by the centromere and the blue chromosome is also split by the centromere. How is the centromere being split? Because the spindle fibers contract. The spindle fibers arising from the centrioles contract, splitting the centromere, thereby the chromatids separate and move to the opposite poles. Next, we have the telophase 2, where the spindle fibers disappears. We are having only the chromosomes at the poles, which are the daughter chromatids, are at the poles. What happens? Reappearance of the nuclear membrane surrounding the daughter chromatids, reappearance of the nucleolus, and already there are the centrioles present. Only the spindle fibers disappear, the centrioles are still present. Only reappearance of the nuclear membrane, reappearance of the nucleolus at the two poles. And look at the state of the chromosomes, we are still in the anaphase stage. Clear? Next, as I told you, telophase should have early and late. The difference between early and late is with respect to the chromosomes. So, this is the early telophase. We know describing of telophase is reappearance of nuclear membrane, reappearance of nucleolus around the poles, which are surrounding the daughter chromatids, which are further called daughter chromosomes. Again, we are not going, once the nuclear membrane surrounds, they are called daughter chromosomes, and already there is the centrioles. Now, what happens? This is early. Now, in late telophase, what happens is, it's happening with respect to the chromosomes. The chromosomes elongate to form an entangle. So, this chromosome elongates to form an entangled mass, and the red also elongates to form an entangled mass. So, we have the chromosomes in the form of chromatin threads. So, the difference between early and late is with respect to the chromosomes. The chromosomes elongates to form chromatin threads. Clear? So, I am pushing this diagram here. And then the last, now as we have learned, cytokinesis takes place. Last event is cytokinesis is taking place, that is constriction of the cytoplasm, which deepens and divides the cell into two. So, we get two daughter cells here, 1 and 2, with all the details of interface: nuclear membrane, nucleolus, centrioles, nuclear membrane, nucleolus, centrioles, and the chromatins. Be careful with red and blue. So, we got two daughter cells here. Now, this arises from A cell, A daughter cell. So, the same thing should happen to B. See, if you recollect from where we started, we proceeded with A. There were two daughter cells produced as a result of interkinesis, or as a result of meiosis 1, there were two daughter cells, and I named them A and B, and we saw the path of A. Similarly, the path of B is the same. It is passing through the same phases, and finally we are getting two daughter cells with nuclear membrane, nucleolus, centrioles, and the chromosomes, remember, red and blue color. So, finally, as a result, the resultant of meiosis, the end product is 4 daughter cells. So, it matches with the distinction between mitosis and meiosis. Ultimately, how many daughter cells are produced as a result of meiosis? 4 daughter cells. And what is the chromosome number of each daughter cell? It is 2. If you cannot identify from here, go back, you can trace it from here. It is 2. So, 2 chromosomes. So, the chromosome number becomes half. I hope you have understood meiosis too. It is exactly similar to mitosis, provided you continue with chromosomes, 2 chromosomes, and use the red and blue color. What I am going to reiterate or take a recap is only with respect to meiosis 1, which is new to you. So, once again, meiosis 1 is a very lengthy process because it involves a number of stages. A quick recap of the different stages of meiosis. Meiosis 1, meiosis 2, meiosis 1 is divided into prophase 1, metaphase 1, anaphase 1, telophase 1, and meiosis 2 is divided into prophase 2, metaphase 2, anaphase 2, telophase 2, and prophase 1 is divided into leptothene, zygotene, pachytene, diplothene, and diakinesis. Now, moving from interphase, we know the typical interphase. The nuclear membrane is intact, nucleolus is intact, centrioles outside is also intact, but the chromosomes are in the form of fine threads called chromatin thread. Now, it is going to prophase 1. The first phase is leptothene. Leptothene, what happens? Condensation of the chromatin to form rod-like structures called the chromosomes. Then zygotene, what is happening? Pairing of the homologous chromosomes. And this pairing of homologous chromosomes, rod-like chromosomes, is called synapses. So, we have 2 bivalents, one red bivalent and one blue bivalence. Then in pachytene, 2 events are occurring, C and C. Again, further condensation such that the rod appears egg-shaped. So, the bivalent will appear now as tetrads, and not only that, crossing over takes place. What is crossing over? The overlapping or it is a crossing over between 2 non-sister adjacent chromosomes. Okay, such a way that they form egg-shaped structures called called chiasmata. So, crossing over is the event that is occurring in pachytene stage. It is the exchange of parts between 2 non-sister adjacent chromosomes of the tetrad to form an egg-shaped structure called the chiasmata. Diplothene, what is happening is the chiasmata breaks and the chromosome separates with the exchanged parts. And diakinesis, what happens is nothing with respect to the chromosomes, only the nucleolus and the nuclear membrane disappears. Meanwhile, the centrioles have duplicated and reached the poles. Now, metaphase, what happens is the tetrads align at the equatorial plane, and not only that, spindle fibers arise from the centrioles from the opposite poles, which attaches to the homologous chromosomes of each tetrad. Anaphase, as usual, contraction of the spindle fibers occurs, separating the chromosomes from the tetrads, the homologous chromosomes of each tetrad. There was a red homologous with the red, they separate and go to the opposite poles. Same way the blue and the blue, they were homologous, they separate and move to the opposite poles due to the pulling of the spindle fibers. Telophase, what happens is nuclear membrane reappears, nucleolus reappears at the 2 poles, and as a result in telophase 1 there are 2 nuclei in one cell. Slowly what happens, it goes to cytokinesis where the cytoplasm constricts and splits the cell into 2 daughter cells. So, we have 2 daughter cells, and finally within the daughter cells in the cytokinesis you can see the 2 egg-shaped chromosomes, one red, one blue, here one red, one blue. What happens in the next phase called interkinesis? These 2 egg-shaped condensed chromosomes elongates. When they elongate, they form chromatin fibers. This is interkinesis, which is a very brief phase between meiosis 1 and meiosis 2. Then they proceed to meiosis 2, which is equivalent to mitosis. Ultimately, the final product of meiosis is 4 daughter cells, and each daughter cells having half the number of chromosomes of the parent. We started with the parent having 4 chromosomes. Okay, how we started initially is with parents having 4 chromosomes. See, I started with this diagram. Leptothene, where we see 4 visible chromosomes, but the end product we have the 4 daughters with 2 chromosomes. Clear? I hope meiosis is clear, and with that we come to the end of this chapter. Thank you.